phenolic components that constitutes the olive oil. Um, Among the different phenolic components, Oleuropein glycol is the most abundant constituent and has attracted the general attention since Oleuropein glycol shows an interesting multiple pharmacological effect as highlighted in the picture. Uh, Considering its neuroprotective action and the pharmacological profile in general, Oleuropein glycol could be an important drug candidate for treating neurodegenerative disorders, including Alzheimer's disease. In fact, in Alzheimer's disease, amyloid beta fibrils that represent the main hallmark of the pathology, uh, the fibrils are able to induce oxidative stress by their insertion as oligomers into the lipidic bilayer and generating reactive oxygen species causing inflammation and neuronal toxicity. Accordingly, a molecule uh, able to induce an antioxidant response, reducing the cytotoxic effect of amyloid beta fibrils, could represent a promising compounds that could be used for developing effective anti-Alzheimer drugs. Um, in this scenario, oleuropein aglycol in animal model of Alzheimer's disease was found to reduce amyloid beta aggregates, reducing cytotoxicity and inflammation derived from the fibrils. And in particular, was found that oleuropein glycol can act in different ways to produce these effects. Um, it has been demonstrated that oleuropein glycol is able to improve the clearance of amyloid beta by promoting autophagy, but it is not the only effect. It was found that oleuropein glycol can directly bind amyloid beta peptides, preventing its pathological transition from alpha helix to beta sheet. And it was also found that oleuropein glycol is able to interfere with the amyloid beta aggregation, and in particular, oleuropein glycol was found to cause a significant remodeling of the fibrils and the capacity of the fibrils to form toxic oligomers. These evidences demonstrated that oleuropein glycol can be also classified as an amyloid beta fibrils disruptor agent. Accordingly, this interesting effect can be explored by using computational techniques at the molecular level. And this is the aim of my work. And in particular, uh, by applying a extensive computational procedures, we performed several in silico studies that consist of molecular docking and long-time molecular dynamics simulation. Um, uh, Very briefly, after a system preparation, we performed a blind docking calculation of oleuropein glycol within the amyloid beta fibrils. This kind of calculation allowed us to identify an high affinity binding site on the surface of the amyloid beta fibrils. This region was considered as possible binding site and was explored by a more accurate molecular docking calculation in order to reproduce a three-dimensional interaction model of oleuropein glycone within the amyloid beta fibrils. And after that, this complex of oleuropein glycone and amyloid beta fibrils was used as a starting point for performing a long-time molecular dynamics simulation in order to assess, to evaluate the effect of oleuropein glycone within the amyloid beta fibrils. During the computational study, we considered also a system in which the amyloid beta fibrils were alone in order to obtain a direct comparison of the two systems and in order to highlight the role of oleuropein glycol in this kind of system. And all these steps will be discussed and described in details in the next part of the talk. And starting from the system preparation step, we performed an extensive computational refinement in order to obtain a reliable starting structure for further computational study. First of all, we downloaded the structure of amyloid beta fibrils from the protein data bank, and to the structure we added two residues, glutamine 15 and lysine 16, in order to obtain a beta-sheet complementarity of the U-shape pentamer. This kind of calculation was performed by using quantum mechanics and molecular mechanics techniques. This kind of optimization was found to improve the stability of the complex during the calculation, as already demonstrated also by us. After this first extensive refinement, we performed a further optimization of the resulting structure employing protein preparation wizard protocol implemented in a Maestro molecular modeling suite. This step is crucial to obtain a starting structure in which for example, the protonation state of some amino acid, such as histidine, is correctly reported. And regarding oleuropein glycol, the structure was downloaded by from PubChem database and was optimized using macromodel software. This calculation allowed us to identify lower energy conformer that was then submitted to leak prep application for generating the most probable ionization state or tautomeric state at cellular pH value. After this preparation step and with the system prepared in accurate manner, we performed a first step of molecular docking calculation. In particular, we explore the possibility to of oleuropein glycol to bind the performed fibrils, and for this kind of calculation we use molecular docking calculation called blind docking in order to use the oleuropein glycol as a probe for exploring the surface of amyloid beta fibrils. This calculation was performed by Autodoc software, and the results showed that there are three possible binding sites for oleuropein glycol within amyloid beta fibrils. But given that the Site 1 is able to accommodate a large number of Docker solutions. With favorable binding energy, we consider this site as the most probable binding site of oleuropein glycone within the amyloid beta fibrils. Notably, this site is composed by different residues that are enclosed in the central hydrophobic region of the fibrils, and this particular region was found to be extremely relevant for fibrils assembly and stability. So, after the identification of the potential high affinity binding site of oleuropein glycone within the fibrils, we perform a more accurate docking calculation in order to reproduce a reliable starting structure. For this step, we use glide software using extra precision as scoring function. The output of the docking calculation is reported in the picture, and as you can see, we found that the catechol moiety of the oleuropein glycone is able to form a 2H bonds with glutamate 22, and it is also able to interact by hydrophobic contacts with phenylalanine 20 and alanine 21. The central region of the molecule is able to target by an H bond with the backbone of phenylalanine 19. So the tail of the molecules is deeply inserted into the fibrils, targeting different several residues belonging to different chains. So this pattern of interaction found for oleuropein glycone seems to be extremely interesting, since this motif enclosed by the residues leucine 17 and glutamate 22 is a motif that is well known, and it is important for fibrils aggregation and stability, as reported in different studies. And this complex obtained by glide score showed a relevant computational score, and so the energies found for this interaction is good, and it seems plausible that this polyphenol could have a pattern of interaction like this within the surface of amyloid beta fibrils. And this complex was then used for investigation the effects of oleuropein glycone within the preformed fibrils. And for this purpose, we used molecular dynamics techniques called long-time molecular dynamics, using Desmond software, and for this computational approach, the software was combined with different parameters such as the OPLS force field and other parameters for simulating the physiological condition. Accordingly, after the setting and the calculation, we calculated one trajectory for each complex, oleuropein glycone and amyloid beta fibrils, and amyloid beta fibrils alone, of 5 microseconds for each system. And by using this kind of simulation, it's possible to capture molecular events that is not possible to observe with classical molecular dynamics studies. And in fact, as you can see, the results obtained by this long-time molecular dynamics simulation uh, highlighted the role of oleuropein glycone as amyloid beta fibrils disruptor. And in particular, the molecule seems to cut the preformed amyloid beta fibrils, being able to induce a strong destabilization of the system starting from about 2 microseconds. For the rest of the simulation, as you can see, the molecule goes deeply into the fibrils, targeting the hydrophobic region principally, causing a total disaggregation of the fibrils. And notably, this action, in addition to cause a complete disruption of the order structure of amyloid beta fibrils, can reduce the possibility to form toxic oligomers. And moreover, very interesting, we observe also a formation of alpha helix motif that seems to be induced by the of oleuropein glycol, and a reduction of beta sheet content as calculated by this tool. And considering that the starting beta sheet content is over 76%, and after 5 microseconds of simulation in presence of oleuropein glycol, we found that the beta sheet contents is dramatically reduced to 12%. And while the formation of the alpha helix motif allowed to detect alpha helix motif from 0% in the starting structure to 16% after 5 microseconds. The reduction of the beta sheet content is a crucial step for reducing the cytotoxicity induced by amyloid beta fibrils, and also the reduction of toxic oligomers and toxic motif containing beta sheet motif. For highlighting the stabilization of the system, we also calculated the root mean square deviation and root mean square fluctuation. And this data further demonstrated that the presence of oleuropein glycol is highly critical for producing a strong destabilization of the amyloid fibrils. And these results were also observed by the timeline representation of the interactions and the contacts of oleuropein glycol within amyloid beta fibrils. And this kind of analysis highlights the movement of oleuropein glycol within the fibrils, and starting from the chain A, oleuropein glycol is able to destabilize amyloid beta fibrils by targeting the motif comprised by leucine 17 and aspartate 23. In addition, during the molecular dynamics simulation, we found other region in the amyloid beta peptide that are targeted by oleuropein glycol. In particular, the tail of the peptides that is comprised from by residues 39, 42. This uh, targeting is relevant since this region was found important for fibrous assembly and stability. So after the first interaction with chain A, the molecule is able to go deeply into the fibers, reaching chain E after about 2 microseconds to 3 microseconds. So this pattern of interaction was directly compared with the results obtained in the long-time molecular dynamics simulation of the amyloid alone. So starting from the structure of amyloid beta and observing the trajectory for 5 microseconds, we observe that there is a general stability of the system and no destabilizing uh, with exclusion of a slight rotation of the pentameric structure for reaching a well-known conformation, namely S shape. This conformation was also observed by other researchers when the amyloid beta is alone into a system, into also in physiological condition. So this computational data is in agreement with the experimental one that demonstrated that this chain of conformation from the U shape to S shape. Also, the calculation of the secondary structure elements show that the contents of beta sheet is maintained high, and no formation of alpha helix was found. In fact, the percentage of alpha helix motif detected during this 5 microseconds of simulation remain zero. And while the beta sheet content uh, that is that was 76% in the starting structure reach about 43% at the end of the simulation. And these contents of beta sheet and the order structure that is not left during the simulation could allow the role of amyloid of the Oleuropein aglycone as amyloid beta fibrous disruptor, since this conformation of amyloid beta is still toxic for the cellular environment. And so comparing the two simulations is also possible to perfectly highlight the destabilizing role of oleuropein glycone within the amyloid beta preformed fibrils. And for assessing this event also by calculating the root mean square deviation, we perform this kind of a calculation for all the chain in the pentamer, and we found that the stability of the system is reached after 3 microseconds, and the systems goes towards general stability and without the introduction of perturbative events like the addition of polyphenol to the system. And this investigation, in summary, we have presented the latest work in which we developed a computational protocol based on molecular docking and molecular dynamics simulation in order to elucidate the mechanism of action of Oleuropein glycone as amyloid beta fibrils. This destabilization of amyloid beta fibrils could be ascribable to the targeting of the central hydrophobic region of the fibrils that is relevant, as previously mentioned, for fibrils assembly and stability. This mechanism of action allowed to reduce the formation of amyloid beta aggregates and toxic oligomers. So this interesting action of oleuropein glycone is potentially useful to reduce the oxidative stress associated with Alzheimer's disease and to plaque loads. In conclusion, our work could be relevant since it provides the guidelines for investigating nutraceutical products, food components, natural products for a prospective application in controlling neurotoxicity and oxidative stress, mainly in Alzheimer's disease. And finally, I wish to thank my collaborators for their active support within the work and you for the attention during the lecture. Thank you very much. Obviously, if you have some question, I will be happy to answer.
Thank you, Simone. So now we'd like to invite the students and our colleagues to ask questions. Okay,
Yes,
so you may post your question on the chat box or
yeah, yeah, yeah.
the host can
Perfect.
unmute.
No problem. No problem.
Okay,
Okay.
we have a question here from Mikhail Harvey Kabunok, a medtech student from the Faculty of Pharmacy. And he asked, since in silico methodologies, particularly molecular docking, gives only relative values for binding affinities, was there a control compound that was compared against the results of the experiment?
This is a good question, since molecular docking is not particularly able to correctly rank the compounds based on the binding affinities. So there are a lot of studies about the possibility to compare the experimental data with the molecular docking results. In particular, we use the different kind of compounds, starting from our previously developed compounds that were able to interfere with the aggregation of amyloid beta fibrils and able also to disaggregate the performing fibrils. So in the case of our in-house compounds, we have in our end different binding affinity to amyloid beta fibrils, and we used that compounds as a reference for assessing the role of fluoropena glycone. This is the step. Moreover, we have also We have also calculated the relative binding energy using prime MMGBSA method for assessing the relative binding affinity. But we have performed a comparison with in-house compounds that bind the amyloid beta fibers with experimental activity. So we have evaluated the calculation derived from Oleuropein aglycone with the experimental data derived from our previously experiments.
Okay, thank you. So there's another question here posted by Mr. Delphine Inigo Pilapil. He is a medical biology student. So he wants to ask, why is blind docking preferred over individually targeting the three sites? And second, which score is more superior, the glide score or the autodoc vena score?
Okay, thank you for the question. We performed the blind docking calculation since we had not indication about the possible binding site of oleuropein glycone within the amyloid beta fibrils. So autodoc is really useful for performing this kind of calculation, providing complexes of interaction, also without uh, any previous indication of the binding site. The blind docking was selected in order to identify the three sites that I have showed, and the score regarding glide and autodoc were similar, and with very few differences. In the order of differences is under one kind of kcal on mol. So a very slight difference.
Okay, other questions? From Joshua Santos, he is a faculty member of the Faculty of Pharmacy. He's asking, Alzheimer's disease is described as a multi-complex disease. Your study only targeted one of the hypotheses, which is the amyloid hypothesis. Would you consider? Would you would you like to consider also other enzymes targeting other hypotheses regarding Alzheimer's disease?
This is also an interesting question. Sure, surely the Alzheimer's disease is a complex disease involving different features, and we considered natural products very important and interesting, since for some of natural products or food components it is possible to highlight a variety of the pharmacological effects. I have showed only the anti-amyloidogenic profile of this polyphenol, but other studies highlight that oleuropein glycone possess other effects on central nervous system, in particular antioxidant effect that could be not derived from this kind of action, but targeting other pathways. So oleuropein glycone possesses a large range of effects, and it is also able to promote the clearance of amyloid beta by promoting autophagy. So all these mechanisms that are found for oleuropein glycone could be very important for developing an effective anti-Alzheimer drugs. In other words, we need to target different pathways for developing an effective anti-Alzheimer drugs. And oleuropein glycone is able to target different pathways, including the amyloid pathway.
Okay, here's another question from Robert Riggs Rondilla. He is a research fellow from the Department of Science and Technology. Um, his question is out of his curiosity. Since docking studies are done on computers, is there any effect on the docking score or results when his computer has limited memory? More so, what are the recommended models of a PC for molecular modeling?
Uh, Thank you for the question. No, in general, the limited memory of the computer is not affect the results. It is only affect the patience of the user, since the time required for the calculation is longer than with a more with a very very with a PC with a large memory. But in this case, the calculation, especially in the molecular dynamics on the GPU with the graphic cards. So the time required for the calculation was important, but not so long, considering that for performing 5 microseconds of simulation on two graphic cards, we have waited 10 days about of calculation for each system, but after that there is the analysis of the trajectory that is complicated as the setting for the calculation.
Okay. Thank you. So there's another question from Sir Luis Mortel. Since amyloid studies are popular for AD research, I'm just curious how well known the LBFF AED motif is so far. Oleuropein is a rather small molecule, and I can imagine other natural molecules with similar structural features. Do you think that there are specific natural product class that have greatest potential for the same site or
Yes,
binding site?
good question, and the answer is yes. The motif was established from some years ago, 10 years, I believe, I don't remember, but it is was found that this motif was important in fibrous assembly and stability. It was found that by biophysical experiments principally. After that, the computational methodologies help in understanding the role of this motif. But experimental data highlighted that this motif is extremely important for fibrous assembly and stability. For the second questions, I believe yes. I believe yes, since more polyphenol, including turmeric compounds, are able to target the same region. So I believe also the other compounds that have a similar action of Oleuropein aglycone was found to target this region. So accordingly, it is possible that polyphenol compounds derived from natural source could be a class of compounds that is able to target this kind of motives. So further studies are required to enclose chemical structural classes, into this targeting. But it is possible that, for example, polyphenol compounds derived from food, from natural source, are able to target motif producing a similar effect for example, the turmeric compounds.
Okay. Here's another question from Jeremiah Gabriel Africa. He is a third-year chemistry student. He wants to ask if instead of directly targeting the amylose fibrils using the compound, is it possible to use the compound to target the precursor compounds of amyloid fibrils instead?
There are not work in this sense, since it is more difficult to investigate the previous step of the formation of amyloid beta. So amyloid beta was formed starting from the amyloid precursor protein that is cut at different level from the enzyme called secretase. There are a line of research that is involved in targeting this kind of enzyme for reducing the formation of amyloid beta in its final form. But if I am not wrong, I believe that some clinical trials with molecules, with synthetic molecules that were able to target beta secretase in particular were withdrawn due to the toxicity derived from the use of these compounds targeting that pathway. So it could be interesting if compounds also possesses this kind of feature. But this line of research is still on, but the results are not very interesting.
Okay. Here's another question from Matthew Denuel Herrera. Since he saw that some of the benefits of olive oil is almost synonymous with antiviral properties of coconut oil or extra virgin coconut oil, would it be possible to find some properties in locally available coconut oils as they both are composed of unsaturated fats?
It is possible. It is possible, depending of the composition of coconut oil. Clearly, I am involved in investigating olive oil since it is a product of my country, and it is a pivotal component of the Mediterranean diet. And so I have investigated, and I'm continuing to investigate the properties of olive oil also in other contexts, since it is a product of my my country. So it will be possible that also other oil possess interesting properties, and in particular you need to assess the exact composition of coconut oil, and if there are some phenolic components, and after that these phenolic components could constitute interesting source for the application on neurodegenerative disorders, since unsaturated fats was found extremely important in neuroprotection, also that in targeting amyloid beta. So the unsaturated fats are extremely important, and they are a key component of the diet that could have a protective role, for example, in neuroprotection, but it is not the only effect of unsaturated fats. So it is possible in general, it is possible.
Okay, so here's another question from Bon Novi de Leon. He is a medical biology student. Was the pharmacokinetic profile, specifically the drugability and blood-brain barrier permeability of OA, tested in silico? What possible solutions could be employed if OA is not druggable?
Okay, sorry, Professor Macabeo, but before I read another question that I can answer now, in which the question is, I ask why RMSF and RMSD calculations are
Yes, sorry.
vital in studies such as this. The calculation about root mean square deviation and root mean square fluctuation are extremely important, since by these calculations it is possible to observe the stability of the system. And so by this calculation, we have a quantitative measurement of the stability of the system. By observing the trajectory and by reporting some step of the trajectory, we can observe the events in the macroscopic area. But in details and quantitative manner, we can assess the trajectory by calculating these parameters. So the comparison of the parameters belonging to different system, as reported in the talk, Oleuropein aglycone and amyloid beta, and amyloid beta alone, gave us a clear picture of the destabilization performed by Oleuropein aglycone. So the comparison and the calculation is absolutely important and, for the molecular dynamics study. So the second question. Regarding the Oleuropein aglycone, it is well established that it is able to cross the blood-brain barrier, since it is also administered by in vivo, and so the pharmacokinetics is quite good, but it's not so good, not in terms of the crossing of the blood-brain barrier, but it is limited the absorption and distribution of the molecule. So the oral administration of fluoropinaglycone as a component, it is not the good way of administration. Also, in the in vivo studies, the oleuropein aglycone was administered intraperitoneally. It is complicated since a very large number of natural compounds, including turmeric compounds, have problems of bioavailability. The research is searching for different methods of delivery of this kind of compounds that have an interesting pharmacological profile, but they have a limited pharmacokinetics profile, and the solution could be, for example, as for turmeric compounds, to enclose the molecules into some lipidic cells, microparticles, or so on, for improving the bioavailability. At the moment, it is very difficult to predict the fate of the natural products like oleuropein glycone, but there are different methods for improving the delivery of natural compounds.
Okay, to ask the last question, may we call on Dr. Albano, our chair?
Hi again. Hi, Simone. So again, thank you very much. Grazie. And my question is, how would you compare the treatment of Alzheimer's? Because I know the current is using the cholinesterase inhibitor. How would you compare it with the amyloid treatment?
The best the best profile for a potential anti-Alzheimer drugs could also comprise the inhibition of acetylcholinesterase, and but it is not in my opinion, the crucial hallmark to target, since as you know, the only approved drugs target this kind of system, but the effects are only symptomatologic. For example, donepezil is not a disease-modifying drug. So we need to search compounds that are able to revert the phenotype of Alzheimer's disease, and not to use compounds or drugs only for the symptoms. So the research should be go on this aspect, in my opinion. But it is not an easy way, and for this reason that the investigation of natural products is absolutely important, since the food components could be well absorbed, for example, and could produce several benefits on our body that could be in line with the profile of potential anti-Alzheimer drugs.
Thank you. Thank you so much.
Okay. Thank you, Simone. There's a last question. I cannot say no to this guy because he is instrumental to contacting you initially, no? So Kin from Kin Israel Notarte from the Faculty of Medicine and Surgery wants to ask you a question.
Hello, Professor Simone. Good afternoon.
Hi, Kin.
Congratulations for that wonderful talk. So this is my question. So has
Thank you.
there been 3D QSAR simulations performed for oleuropein aglycone? And what functional groups in OA that might have contributed to its stable interaction with the amyloid fibril? And will it be possible in the future to produce several derivatives of OA to determine which has the best anti-amyloid property? And since you mentioned also a while ago that in terms of its blood-brain barrier crossing capacity, it's quite not good. So perhaps no one of this derivative would be able to enhance its ability to cross the blood-brain barrier, particularly that the amyloid fibrils that we are targeting is located in the brain. So of course, for the drug to work, it really has to pass through the blood-brain barrier. So that's it, Professor.
Thank you, Ken, for the question. So, in this case, the three-dimensional QSAR based on a set of polyphenol, for example, that were able to target the amyloid beta could be interesting. And based on the similarity of the structure, for example, of turmeric compounds or other derivatives that showed this kind of action. Probably the catechol moiety and in general should be important for targeting the polar area of the mentioned region, but the hydrophobic contribution of the rest of the molecule seems to be also very important. And probably the activity in this sense, it is a um, summation of different features into the molecules, in particular catechol moiety, but also an hydrophobic contribution in the molecule. It is necessary to interact with the hydrophobic region. So it is not really easy to highlight crucial groups in this kind of molecule, since, in my opinion, is the molecule, not the features of the molecules. But regarding your second question, it could be really interesting to develop a subset of derivatives for evaluating the possibility to improve the physicochemical properties, the ADMET profile, in order to provide compounds that can derive it from natural source, but with improved properties. And it could be very interesting to explore. And if you have some compounds, I am ready to study these derivatives, or we can also think to develop some compounds in order to assess the possible role in Alzheimer's disease. Why not?
Thank you very much, Professor. That was a wonderful talk.
Thank you.
And thank you also for opening up for possible collaboration.
Yeah.
Thank you.
Thank you, Ken. Professor, you have the mic. Okay.
Yeah, because I was muted by the, yes. So thank you again, Simone, for answering all the questions, curiosities of our students and colleagues. And I would also like to thank the students and guests, okay, guests as far as guests from the UP, University of the Philippines in Diliman. So the RAs of Dr. Ricky Nelias. Also, there's a faculty from the De La Salle University and also research fellows from the DOST, and also Warren, my former student who is now a PhD student in the University of North Carolina. So he also attended this meeting. Of course, I'd like to thank our colleagues in the Department of Chemistry and the chemistry students for participating, and also the medical biology students, one microbiology students. No, thank you. Also, our noteworthy to mention our friends from the Faculty of Pharmacy, from the Department of Biochemistry and Pharmacy and Medical Technology, no? So special
Also the
mention to
psychology students.
yes, sorry. Okay. And especially to my advices from the faculty of from the Department of Medical Technology, who is now doing some molecular docking studies with COVID spike proteins, no? And the faculties of the Faculty of Pharmacy. Okay. So let me now call again our chair to present our token of appreciation.
As a token of appreciation, Dr. Simone, we would like to give this certificate of appreciation awarded to you as to Assistant Professor Simone Brogi, MSc, PhD, for delivering a webinar entitled Natural Products as Sources for Potential Anti-Alzheimer's Drugs, the case of Oleuropein, aglio, it's too small, so I can't read, aglycone, given this 27th day of November 2020 at the University of Santo Tomas, Manila, Philippines, signed by yours truly, the chair of the Department of Chemistry, and also the Dean of the College of Science, Professor Raydon S. Papa, PhD. Thank you so much, Simone. And I
Thank you
hope
very much.
to we'll have another series of webinar, maybe for COVID or other diseases that you're working on. Okay.
Yeah. Thank you. I will be happy to participate to other webinar.
Yeah.
But thank you for the invitation to this webinar, and thank you very much. Thank you.
Okay,
Thank you.
so to our audience, can you show your appreciation to Assistant Professor Dr. Brogi by taking the reactions emojis? Okay, and now I would like to call our beautiful, fresh Assistant Dean of the College of Science, Professor Maria Claudette A. Agnes, to give her closing remarks.
Good afternoon, everyone. Good afternoon, Professor Simone Brogi. Good afternoon to our very energetic department chair, Dr. Albano, Dr. Macabeo, and all the Department of Chemistry faculty members, the students who participated in our webinar. Thank you so much. I would like to commend the department for selecting the topic on Alzheimer's disease. You know, I was listening this afternoon your talk, Professor Brogi, and I am very much interested. And you all know that in the College of Science, we have different departments. We have the Department of Biology, the Biological Sciences, Department of Chemistry, Department of Psychology. And we are very much also interested with your talk, especially that right now there is no specific drug for Alzheimer's. And I was listening to your talk and recalling about the natural products, no? And with the natural products here in the Philippines, we have the coconut oils. And you mentioned a while ago there was a question regarding that matter, and probably that would help. And with this, we are very much aware that later on, the Filipinos will be helped regarding the problem of Alzheimer's. It's not only in Europe, it's not only in the States that Alzheimer's is dominant. Here also in the Philippines, we have a lot of patients. And we all know that Alzheimer's disease is an irreversible, progressive brain disorder that slowly destroys the memory and thinking skills and eventually the ability to carry out the simple tasks, no? So I hope what you have discussed this afternoon will be applied by our participants. Thank you so much. And I hope that next time, if we will invite the College of Science, particularly the Department of Chemistry, you will again very much willing to give a talk and collaborate with our researchers on Alzheimer's disease. Probably Dr. Albano, Dr. Macabeo, we can ask faculty members from different departments, no? Because Alzheimer's could be a research on Alzheimer's disease could also be collaborated by different departments. We have mathematics and physics, we have biological sciences, chemistry, and psychology. So thank you so much. And once again, good day to everyone.
Thank you, Assistant Dean, for that wonderful closing remarks. Before we tick the leave button, guys, the department requests that you fill out the evaluation form for today's seminar, and you can see on the screen the QR code. So kindly fill out the necessary informations, no? So again, thank you, Dr. Brogi, for gracing the Department of Chemistry's webinar series in drug discovery. And to all participants and colleagues who also made it for today's meeting. Thank you, and it's still good afternoon, no?
Thank you and good afternoon to you.
Yes.
So let's have a photo op. Let's not
Yes.
forget. Yeah.
Yes.
Don't leave yet.
So, who is in charge?
Melville, yeah. So may I ask everyone, students, faculties, to open their cam and join us with a photo op. No? Yeah, I'm happy to see all the faculty members and all the students who join us.
Yes, we have 232.
232 at most, yeah.
Yeah, that's right. So that's a lot of data.
Yeah. Good luck, Zoom.
Okay, first slide, first set of picture, next set.
Smile.
Next set.
I think the first set can now relax.
Okay.
Yeah.
Was I included in the first set? Okay.
Yeah, I think we're in the first set. Yeah.
Okay.
Okay, next set.
I think that's a lot of sets.
Yes, stay
Yes,
put.
9.
Yeah, I see Dr.
Hi,
Tongo.
ma'am.
Yeah. Hi, Dr. Tongo. How's your quiz?
Okay, 5th.
Who's taking the photo?
Me po, Aldrin po.
Okay, Aldrin. Sige.
Yeah. Ken,
6.
I see Ken also. Ma'am Christy. Yeah. Hello, Ma'am Christy. Smile.
Hello po from Ilocos.
Sir Bergs also. Uh,
7.
Thank you for attending. I know you have a class later at 6 p.m.
Okay, last set.
There are 9 sets, I believe. Yeah.
Yes po.
So
Okay, so already, I took already all the pictures.
1 micro 1. Hello.
okay.
Sir Aldrin, I took care for the last set.
Okay. Thank you so much, everyone.
Bye.
And see you again in the next webinar of the Department
Yeah,
of
that's
Medicine.
on
you.
Wednesday.
Wednesday, December 2, 5 p.m., I believe, Philippine time.
Yes.
Yeah. Okay.
Bye. Thank you.
Okay. Bye-bye. Thank you.
Thank
See
you,
you
Bye.
Lan.
again.
Thank you,
Thank you,
guys.
Lan. Thank you,
Thank
sir.
you,
Thank
Thank
you.
you,
Simone.
Ted.
Bye-bye.
Bye.
Bye-bye.
Thank you, sir.
Thank
Bye.
you, Audrey.
Bye.
Thank you,
Thank you,
Professor.
sir.
Thank you,
Thank you,
Chris.
Thank
sir. Bye po.
you.
Thank you,
Thank you
Professor
po.
Brogi.
Thank you.
Thank you, sir. Bye.
Po.
Thank you, sir. Thank you very much.
Thank you, sir.
Thank you, Alan. I know you're still out
Welcome.
there. And Alan Salcedo. Thank you, Alan
Sakto
Macabeo.
nakauwi na ako. Just arrived home.
Thank you, Assistant Dean, Ma'am Claudette. Okay.
Leave na? Okay. Uy, thank you sa organizers. Thank you for the eval, the platform,
Thank you.
program.
Thank
Uy,
you.